So photosynthesis. Well, we have to start this discussion, and you might want to take notes for sure. I'm going to give you pretty much everything you need to take the next 2 quizzes. Oh gosh, great news. I excluded quiz number 2. It was horrible. I don't know if you've taken it before or if you've taken it already, but it was the history of photosynthesis discovery, which is absolutely over the top ridiculous. And I checked your final for Apex, your unit test for Apex, and it's not on there. There were maybe 1 or 2 things on there, but they're picked up in quiz 1 and 3 that you might need for AP. Believe me, I'm not going to let you miss that stuff, but that quiz was ridiculous. So it's excluded. You're welcome. You don't have to take quiz number. So quiz 1 and 3 is what we're going to get done today. And then next time we meet, we'll meet and go over the labs, the 2 photosynthesis labs that we need. And then the next time we meet, we'll go over cell respiration, and that'll be the end of the unit. Okay. Cool. That's 2 weeks to the end of the unit. I'm trying to do it 1 unit per month. All right. So the first thing you need to know is that photosynthesis and cell respiration are all about free energy. Now you might see a lot of free energy mentioned here and there in the Apex curriculum, but AP Bio, the College Board, now Apex curriculum, let me just explain, is a little bit behind. They haven't done the redesign from 2 years ago yet in their curriculum, so they still have quite a bit about free energy that pops up when we don't have it at all in the curriculum that we're doing in the classroom. So it was taken off the formula sheet 2 years ago. But still, it's not bad to know it, because you're going to have it for your Apex curriculum. So what it means by free energy is available energy, energy that is available for use in systems, and we're looking at biological systems. So anytime you see free energy, you can just think available energy. Okay, how is the energy, what's available? And so that's what it means by free, like it's available, okay? Of course, there's laws of thermodynamics that state we can't destroy energy, and we can't just create energy out of anywhere. It's basically a recycled effort. But what you need to know for photosynthesis is the free energy that and sorry, I'll try not to do all my weird abbreviations. We don't know each other that well yet. I do NRG for energy, but anyway, free energy largely comes from the sun, okay? In photosynthesis, or really, I guess if you wanted to take it back to the big picture in all of our biological systems, but we're talking about photosynthesis today. So whenever you're wondering, oh gosh, well, where does this energy come from? The free energy that we are using in photosynthesis, the energy that's available for us to use in photosynthesis, and I say us as if I'm a plant. I don't do photosynthesis, but that comes from the sun. Okay? So that's the first thing that you need to be aware of. We're going to get this energy from the sun, and I'm going to show you how it's done. This is going to be kind of a really fast walk through it, and so don't be alarmed if at the end of it you're like, holy crap, I don't even know what I just learned. That's okay. You'll have a total list. You'll have the picture. I'll try to explain it, and then you can go back and kind of do some of your reading to fill in some of to kind of reinforce it, because it is going to be a lot. Okay. So our free energy that we have available comes from the sun. We have 2 processes in biology that we use for energetics. This is what, energetic stuff. One of them is photosynthesis, and that is in the chloroplast. So that is, and also my notes are not ever very organized. I apologize. I'm kind of scattered. So in the chloroplast is where photosynthesis occurs. This is in autotrophs. And you want to be very careful assuming that's only plants. It's not only plants. Okay. It's plants. And so I'm going to draw a little line here. Plants, of course. It's also, there are some, I'm going to put some because it's important, it's not all, some protists. Okay, protists are, you have 4 kingdoms of eukaryotes. Eukaryotes are organisms that have a nucleus. There's 4 kingdoms of them, right? There's plants, animals, fungus, and protists. Protists are what I call the junk drawer of the eukaryotes, because we have no idea where half of them go. We just slap them all in that drawer together because I don't know what it is. But some protists are autotrophic, photosynthetic. They have chloroplasts. They can do photosynthesis. So these are things like some of our algae. Gets more complicated. Some of our algae are classified as plants. But anyway, some of our algae and mosses, things like that, can be photosynthetic. And also there's some single-celled organisms that are protists that are also photosynthetic. So that's kind of cool. And then, of course, this is the big one. So we have some prokaryotes, and not all of them. This is the big one, and I say big one because this is where photosynthesis is hypothetically or theoretically, of course, we weren't there when it happened. So theoretically, photosynthesis originated in prokaryotes. Remember, prokaryotes have no organelles, so no chloroplasts here. So they have to do photosynthesis and cell respiration on their membrane, on their like cell membrane. Okay, we'll talk more about that really when we get into cellular respiration. Okay, so today our focus is going to be on photosynthesis, but you do need to be aware that we take the energy that is created, and by we I mean all organisms take energy that is created or is stored in photosynthesis, and we use it in our mitochondria to do cellular respiration. Okay, so that's kind of what the flow is. There's a couple of questions on these first quizzes that you're going to have to do that talk about cellular respiration and mitochondria. It's not the focus, but there's like a little overview paragraph thingy or a section in the beginning that talks about the two sources of energy, the two energetics pathways that we do in biology. And so just know, and you can write down if you want, but I don't have a spot for it here. I'm doing photosynthesis right now. Just know that mitochondria is where cellular respiration happens. And that's when we take that sugar that's made in the plants and convert it into energy that's usable for our cells. Okay. Because they ask a question about like, what would happen if the mitochondria failed or something, and it would be the cellular respiration would fail, but not photosynthesis. Okay. Good so far? Questions so far? Thanks for the thumbs up. Okay. Alrighty. So that's the kind of the beginning story. And I do have my list over here to make sure that I hit everything. So let me check and make sure I don't have to hit anything else before we get into the nitty gritty of cell respiration. Okay, cool. All right, I'm going to erase. Anybody need more time? Cool. Okay, so let's talk about photosynthesis. So first of all, photosynthesis is, like I said, only for autotrophs. So these are organisms that can create their own food. Well, that's pretty cool. And we're going to go with eukaryotes here because that's where we're going to focus this whole entire little lesson. But just know that prokaryotes are where they feel like it originated. So bacteria is where they feel like it originated. And also that they do this without a chloroplast. Okay, so I'm going to zoom in on a chloroplast. So I do a lot of drawing, and you'll have to just forgive me because my drawings are usually pretty ugly, but they get the point across. Okay, so we need to know kind of the structure of a chloroplast, where it's located, and some of the stuff in it in order to do this. So let's get a little anatomy here. Okay, so this typically takes place, you're going to find this in the green parts of plants. And I'm going to say, I don't even care if it's plants. I'm going to say autotrophs because that makes more sense. Because it could be plants or it could be protists. Mostly plants for us. And of course, eukaryotes do a lot of photosynthesis. Okay, so in the green parts of autotrophs, so basically you're not seeing this in fruit, right? Apples, potatoes. You're not seeing this in onions, like the bulb of it. Of course, the little green stem that comes at the top, you're going to see that's going to have chloroplasts in it. But you're looking at the green portions of the plant. Okay, so the leaves, really the leaves. The leaves are awesome solar panel, right? Because they're super broad, and the more broad they get, you can think they're in a nice warm environment that's got lots of sun. They're just soaking up the sun. Okay, and that's really what they're for. So, and within that, you are going to see, and I mean, I could draw a little leaf here, I guess, if I wanted to. Why not? Why the heck not? So here's our little leaf here. Okay, so a little leaf of a little plant, a little stem. Okay, and so we're going to zoom in on a little plant cell. Okay, and they always draw them like boxes. They don't have to be boxes, but they're pretty geometrical. So it's a plant cell. And you would have like, and I have a GIF of one. Oh, well, I don't feel like changing my screen. So you'd have like a nucleus because you have organelles, right? You have a membrane around the outside edge. Okay, so you have your cell membrane. You have a cell wall, just reviewing some plant structures here for a second. It's okay. You have a big fat vacuole in the middle. Okay, so this is a vacuole. It's full of water, which is super important to photosynthesis because we need water to do this. This is your nucleus, which I won't be able to label because there's no room. And then where your, oh man, I have a green marker. I have red, blue, black. Oh well. where your chloroplasts are usually all around. So pretend like those are green. If you have a green pen and you're drawing this, then draw them green. Or if you have you want to look up a plant cell diagram, you can just see one on your screen. So the chloroplasts are all around the plant cell. They do something called they do something called streaming, and they move. Actually, if you get really lucky when you're looking under a microscope, you can change the light to one side and you'll see all the chloroplasts just move over to where that light source is coming from. Super cool. But what they're trying to do is collect as much light as they can. Okay, so we're going to zoom in on one of these chloroplasts. So here we go again. See, I told you I draw a lot. So we're zooming in on a chloroplast, and they look kind of like, oh, I don't know, a little flat-bottomed thingy-majigger. Okay, they have a double membrane, which is really strange, by the way. There are two organelles that we have that have a double membrane, and guess what? They are a chloroplast and a mitochondria, both of our energetics ones. So double membrane is important. Now I have something to tell you that's kind of weird. One of your quiz questions has a really strange answer, and I don't like it. So I'm just going to tell you what that question is, and I'm going to tell you what you should answer because it doesn't make any sense with chloroplasts. It asks why a double membrane is beneficial for us, and it says something like so they can build up a proton gradient. And we'll talk about protons and gradients here in a second, and I'll revisit this, but that's not true. That's totally incorrect. That's what a mitochondria does. So that question, I think it's on the second quiz, it'll be the third quiz. Second one is excluded. But on that third quiz, for some reason, it totally misidentifies that, which is frustrating. So anyway, so it says that the reason there's a double membrane is so that the proton gradient can build up, but that's not what happens. So double membrane, I'm drawing some little stacks of coins in here. You probably should either draw or find a picture of the chloroplast to kind of learn what all the parts are. We have to zoom in again, by the way. We're not done zooming. So the deal with our double membrane is that there is a theory called the endosymbiotic theory. Are you familiar with that? No? Okay, cool. Let's be familiar with it because it's one of the most important things in biology. I shouldn't say that. Everything's important. Endosymbiotic theory. And this is where this double membrane comes from. So that's why I want to talk about it right now. Endosymbiotic theory states that this means inside. This means like symbiosis, life together. Maybe you've heard of like mutualism, parasitism, junk like that. So endosymbiotic means that there is a life together inside kind of theory somewhere. Wonder what this is all about. So the theory goes like this. Once upon a time, back in the days of the primordial soup, whenever there was a bunch of sludge in the earth and nothing else, there were some prokaryotes that lived in the soup and nothing really else lived in the soup, okay? Prokaryotes, of course, these are your bacteria. There were some that were larger than others, and they would routinely eat the other ones. I mean, what else are you going to do? So the theory says that at some point, there was a photosynthetic prokaryote. There were prokaryotes that could process energy and make ATP, like with their mitochondria. And some big prokaryote engulfed a littler photosynthetic or cellular respiration one organelle. And instead of eating it, like nom, 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 dissolving it, it actually just like started living inside of it, endosymbiotic. And they started helping each other. And so then the larger prokaryote, of course, would be able to take in nutrients from the outside world. And then the smaller prokaryote would then would be doing its photosynthesis and producing glucose. And so they were like happy together. And that is actually, so the endosymbiotic theory is actually a theory that it tells us really how these organelles came to be inside of our cells. How we started to have like eukaryotes at all. And so that's kind of an interesting theory. However, one of the evidences that this is the case is because of this double membrane. Because if you think of something that has been endocytosis into the cell, so you had a cell and something came along and like was engulfed into the cell, it actually pinches off a part of the cell membrane around it. And so it makes it in like a little vesicle, like a little, traveling pouch. And so it's basically what they're saying is this double membrane, this outer membrane was originally like a cell membrane of a big prokaryote that engulfed it. And so because of that, we end up with an endosymbiotic theory saying our mitochondria and our chloroplasts were once free living organisms. Also, they have their own DNA. They have their own ribosomes. They make their own proteins. They divide on their own schedule. There's a lot of evidence on this with this theory. So it's pretty cool. But that's where the double membrane, they think, comes from, is from being engulfed originally back in the primordial soup days by a larger prokaryote and just becoming like a eukaryotic cell, like an organelle inside of a cell. Questions? Okay, cool. So now moving right along, we are going to zoom in on one of these bad boys right here. So these little things are called a thylakoid. Let me write it out here. Where can you see? I got to check my board. So a thylakoid is one of those little coin-like structures right there. And that thylakoid is basically where photosynthesis happens. Well, also the stroma. I'm going to draw right there because the stroma is like cytoplasm. It's like jelly. Okay, so the stroma is like where is something happens in the stroma too. So we need to know the stroma too. And then the thylakoid is like one of these little stack of coins, what they look like. But they all have like little channels. They're all interconnected. And so there'll be like a channel that's connecting them, whatever. Okay, so let's zoom in on a thylakoid and we'll have some stroma around it so that because it's like surrounded by this cytoplasm. So here is our thylakoid. There it is. It's cute. It has a little membrane too. Okay. This whole thing right here is a chloroplast, right? Everyone know that this thing with the thylakoids in it, that's a chloroplast. So that would be the green part of the plant. That's what's green. Okay. So far, so good? Yeah. When I look off to the left, it's because I'm like looking at your little faces. Everybody good? Okay. Okay. So I'm going to do a little bit of erasing now. So if you want to like make a big thylakoid, you can, because we got to have some more space in here. Like this is not enough space. Okay. All right. I'm going to make an adjustment here. My view. Okay, let's see. Alrighty then. So we are going to look at and kind of an in-depth look. I'm trying to get more of you on my screen because I can only see like 9 of you. So sorry if I'm kind of messing with stuff for a second. Change layout. Oh yeah, it says I can only see 9 of you. Let me bump that up. And you guys, by the way, you have settings on your Google Meet where you can change the layout. Oh, now I can see a bunch of you. Perfect. Now I think I can see all of you, even my ones that are still an icon. You guys suck. Okay, so sorry to be rude. Sometimes I'm a little rude, sorry. Okay, so this is the thylakoid right here. So this is thylakoid where we just blew up. Now it's a kind of oval shape where the chloroplast has a flat bottom. So this is a thylakoid and it's inside. Oh, geez. Pardon me while I write completely off the screen. Okay. And it's inside the chloroplast. So we are deep down inside the cell. Okay? Teeny, teeny, teeny. You're never going to be able to see this in any sort of microscope that you'd have in a high school lab. You might have an electron microscope someday and see one there. Okay, so in this thylakoid, we have, let's pause and write down the photosynthesis formula, shall we? Okay, so what do we need to do photosynthesis? Well, we need some water. Okay. We, of course, need some light energy. We need some carbon dioxide. And if we have those things, and I'm going to balance it in a second, if we have those things, we will make glucose. Please memorize the formula for glucose. There's like no reason not to. Move my thylakoid out of the way because it was in the way. Okay. And we will make oxygen. Okay, I have a story to tell you because, by the way, all these are 6s. It's kind of a devilish formula. So actually, you're going to need 12 waters, and you'll make 6 waters over here, but they just ignore that for some reason. So if you have a full-out formula of photosynthesis, you would see like 12 H2O, 6 CO2 yields glucose, 6 O2, and then also 6 waters. but for some reason they just ignore that. So I don't know what's up with that. That's because in order to create a 6-carbon glucose, you actually have to do this twice. You have to have, well, that's not exactly true. You have to have a lot of water. Okay. So I don't know why they kind of cancel it out. All right. So this is a thylakoid membrane right here. So thylakoid membrane is, and that might be kind of what they were referring to on that one quiz question, but they didn't say thylakoid. They said definitely the double membrane. So that was real weird. And then stroma is out here. So this is stroma, which is like the cytoplasm of the chloroplast. Okay. So that's where we are. And then of course there'd be another thylakoid down here and there'd be a stack of them. Okay. All right. So the first thing we need to do this particular process is we need water. Okay. So water, I'm going to put some water in our cell. So here's some water. And step 1 of photosynthesis is that we have to split these waters because we need the hydrogens. So hydrogens are going to play a big role in this, but they're kind of a fun role. So that we break them apart and we change some charges on them. It's kind of weird. I'm going to put an enzyme here, and actually it's a bunch of enzymes together, but it's called photosystem 2. So we call it PS2. Oh, geez. Can you see that? It's really squishy. So this one's PS2. And it's a system of enzymes and a system of proteins that does some work for us. In photosystem 2, there is chlorophyll. Okay, so I'm going to put it at the top because we're pretending like this is like up and the sun is up at the top. So in this photosystem 2 is chlorophyll. Okay, chlorophyll is a pigment that allows, see if I can get, yeah, there we go. Chlorophyll is a pigment that allows electrons to be excited. So it takes the sun's energy, and when it has electrons in it, it'll like excite them, like bring them up to a higher energy level. Okay, and that's what chlorophyll does. So it's actually a pigment that can do that. It's in photosystem 2 and photosystem 1, which we'll look at in a second. But really, more of that energy exciting part happens in photosystem 2. Okay, so the first thing we have to do is there's an enzyme that's attached to photosystem 2 that you don't need to know the name of, although they do reference it in one of your quizzes. You don't need to know the name of it, but basically what it does is it's a water splitting enzyme. So it splits water, which is kind of cool. We can't usually split water. Water is super stable. And so we can't really usually split it, but there's an enzyme here that splits water. When it splits the water that's brought in from, of course, the roots of the plant, right? Then we have oxygen leaving. Make sure you can see that. You see that? Yeah. So oxygen leaves. And what we have left is hydrogen. Okay. And I'm sorry, it's having a little focusing episode. I don't know what's wrong with my little thing, but we have a hydrogen left. So the oxygens, bye. See you later, oxygens. Of course, they bond together before they leave to make O2 because oxygen is never alone. When we have that, we have a buildup of what we call protons. All right, let's do some chemistry. So protons, so hydrogen is just a proton and an electron. Like, are you aware of that? Like, that's all it is. Hydrogen is a proton and an electron. It's the most simple element that we have. And so if we steal the electron, I'm going to change to blue here. My electron's going to be blue so you can see it better. If we steal the electron from the hydrogen, then all we're left with is a proton. So I'm going to make that's a little E negative. So the E negative stands for electron. Okay, so E negative equals electron in case you're not up on that. Now it looks like a 3 negative sign. There, I'll draw an arrow. Okay, and then H positive is proton. And that's the words we use. Proton, electron, that's what we use here, okay? So even though we write H plus all over the place, we actually call them protons, okay? And then that's an electron. Questions? Okay, cool. So the water is split, the oxygen leaves. This photosystem 2 steals the electron from the hydrogen, which leaves us with a bunch of protons here in the middle of the thylakoid, just hanging out in the middle. And the electron is headed to the chlorophyll to get excited. Okay, It's about to pick up some of that free energy from the sun. That basically is step one of photosynthesis. We good? We call this part of it, we call it the light dependent reactions, because right now we are seeing they have to have light for this to happen, okay? So there's two separate sets of reactions. Can you see that? I think you can. There's a T on the end of that. But anyway, there's two separate sets of reactions for photosynthesis. One is light dependent and one is light independent. Okay, so one happens regardless of light and one has to have light. We're talking about the light dependent. It's the first part of photosynthesis. Okay, so far we have electrons that are being excited in photosystem 1, photosystem PS, I'm sorry, I said 1, PS2, photosystem 2, okay? We have electrons being excited there. We have water that's been split, so oxygen is produced, and we have protons that are building up in the center of the thylakoid. That's where we are right now. It's fixing to get complicated. You thought that was hard. Okay, so here we go. We have something right here that is a series of proteins. I'm not going to draw them correctly because it's like, I don't know, seems to be against my biological religion to draw these correctly. I screwed up every single time, but it's okay. It doesn't really matter because who really cares what they look like? I doubt they look like boxes anyway. All right, so this is the electron transport chain. We have these all over the place in our energy stuff. Electron, Lord have mercy, Miss Wilhoit. They can't even see that. There we go. Electron transport chain. There's massive ones in photosynthesis and there's massive ones in cellular respiration. They do kind of the same thing. Basically, they do the same thing. Okay, so electron transport chains, what they're doing is we're passing these little electrons, which are going to be blue. We're passing these little electrons down the series of proteins, and we're kind of using their energy that they just got from the sun. Like, dude, we just got that, and why are you taking it already? Okay, so that's what we're doing with them. So PS2 has excited and energized the electrons, like got them a lot of energy. They've got lots of energy now. And as we pass them down this, and I always make them going down to kind of symbolize the fact they're losing energy. As we pass them down this particular little electron transport chain, the electron transport chain fires up. It's got proteins, so it's like channel proteins. It's now got some energy, kind of like you'd use ATP to power a protein, but now it's using energy from the electrons. And so what it's going to do is it's going to fire up a little pump, and it's going to pump hydrogen in from the surrounding stroma. I show you. Okay, so I'm going to erase my little bracket here. So we've got a bunch of hydrogens just hanging out up here because they were passed off from the other thylakoid. I'll show you how that happens in the very end. Okay, so when the electrons, it's important, when the electrons go down the electron transport chain, that's like a bucket brigade, like pass from this protein to that protein, to this protein to that protein, okay? When they go down the electron transport chain, it fires up the electron transport chain proteins, and it allows these hydrogens to come through a channel that is an active transport channel, pushing a bunch of hydrogen in where we already have some from splitting the water. So now we have a lot. Okay, so we're making a proton gradient right here in the middle of the thylakoid, which is inside the chloroplast. Okay, we're building up a big proton gradient, which means we have high to low, so that we can move out again. So it's kind of just building up a store of protons so that we can use them in a second. This is where that quiz is incorrect. The quiz asked why there's a double membrane on the actual chloroplast, and said to build up a proton gradient right here, but we're not at the membrane. We're in the stack of thylakoids. So it's very strange. But anyway, just answer it accordingly, and it's fine. Who cares? Okay, cool. Now we have two different directions we could go. We could go into the photosystem 2. Oh my Lord, if I don't say that wrong. Into the photosystem 1, or we can go down to the ATP synthase. They happen simultaneously. So I'm always like, let's finish out this top part first. Okay, so once we get to here, we've got little sad electrons that don't have much energy. They're just kind of chillaxing, right? I'm going to erase some of this. Sorry, I hope your little picture isn't just all crammed, but it probably is. Just draw it again. I make my kids draw it like 10 times. before I'm satisfied that they understand it. And then PS1. Okay, why the heck does PS1 come after PS2? Who was the brilliant scientist that named these things? I can tell you why. They discovered PS1 first. And then when they discovered PS2, by the way, that's Photosystem. They discovered Photosystem 1 first. And then when they discovered Photosystem 2, they didn't want to change its name. So there you have it. Brilliance of science. PS2 is the first one. PS1 is the second one. In PS1, we also have some chlorophyll up at the top. Oh, I didn't draw my little sunshine. There he is. Rays of sunshine. Woo. Powering him up. So the sunshine also powers up there. Of course, we get to re-excite our electrons. Yay! They get re-excited. There they go. I always draw them going down if they're losing energy. They don't really go up and down. That's just kind of my way of like remembering what's going on here. Okay, and so now we have re-excited electrons, but like I said, PS2 does more exciting than PS1, so they don't quite have as much energy as they originally did. And now they get put into electron carriers, and they get carried off, like putting them in a little school bus and taking them on a field trip. Okay, so our electron carriers are over here, and they are NADP+. Okay, that's our electron carrier. Okay, so that means it's got a positive charge, and I don't care if you ever know what this stands for. In fact, I'm going to just come clean with you right now. I don't even know what it stands for. I have to look it up. I don't care. It's NADP+. The one that, there's another one just like it in cell respiration, and I don't care what that stands for either. So NADP+, it's going to pick up an electron, right? And that's going to turn it into NADP, wait for it, H. Our hydrogen is restored because our proton and our positive charge and our electron have been reunited, and now we are a neutral NADPH, and this is the little school bus that's going to carry our electron off to the next step. Okay, I'm going to erase my light-dependent reaction so I can label that for you. So NADPH is an electron carrier. Literally carries electrons, and they have some energy. They're like kind of excited electrons. So they're like, "Woo, let's go do some work." Okay, so that's what they are. Oh, have mercy. Do you have questions? Do you even know what your questions might be? It's just a process, memorizing a process. All right, I'll give you a second. Wait for it till I see like your faces and not like look like you're writing furiously, because we have to finish this out. Remember, these are just the light dependent reactions, the ones that are dependent on light. Sun's up, this works. Sun down, no worky worky. Okay, I'm starting to see some eyeballs and some thumbs ups. I'm telling you, I can't teach if I don't see your faces. I have no idea how fast to go. You would think I haven't been doing this very long. Okay, so then down here is a cool little protein. I mean, Lord, this is the coolest protein ever. It's called ATP synthase, and it literally does that. It synthesizes ATP. I draw it like a, I don't know, like a little bulbous thing, but it actually, dude, Google one sometime. They're so cool. They rotate like a windmill, like a turbine. They do that, and you have them in your cells. It's freaky. ATP synthase. Synthesizing ATP. ACE means enzyme. This is an enzyme that makes ATP. Glory, hallelujah. Let's get some ATP made. All right. What happens now is we have formed a beautiful proton gradient. Look at all those protons. Okay, they are just ready to funnel through this little protein, this enzyme called ATP synthase. They are funneling through there, and when they do, they attach a molecule of ADP, which is adenine diphosphate, and they attach it to another phosphate, a P, and that makes ATP. So I guess I should throw a little P up there too. Usually we do our phosphates like a little circle around them. Okay, so ADP plus P, and the proton gradient from the proton gradient, yes, built up here, moves from an area of high protons to an area of low protons, right? So no energy required. the turbine actually spins as the proton goes through. It's freaking amazing. I really don't know how people figured this out, but it's so cool. And when it does that, just like a water, like a turbine in the water, like a hydro dam, you know, where it makes energy, just like that, it creates just enough energy to bind a phosphate to an ADP, which the cell makes, will recycle, I'll show you in a second, to make ATP, which is a high energy molecule, okay? And now the light reactions are done. Oh, it's just a little bit, right? We only have the light independent to go. Sure, we have 15 minutes. I think I can do it. Okay. All right, let's do it. Let's go through it again. Ready? Walk with me through it really quickly. It's really not that big of a deal. You just have to memorize it. That's what you have to do. So what I have my students do is I actually draw like with a Sharpie. It's really high tech. I draw with a Sharpie like a blank diagram like this. Like I leave out the little parts of it, and then I make a bunch of copies of it, and then they have to sit there and try to fill it in with the step-by-step. Highly recommend. I'll draw one with a Sharpie and put it in your Canvas so you can print it off and try to fill it out. Because until you can do that by memory, you do not have this process. You don't have it. And if you don't have this process, it's really hard to answer the questions about it on the AP test. Okay, so here we go. So in the beginning, we have water. That's the first thing that happens. Step one is water is split. So if you're like just sort of going around and you want to like make a list or something, you can do that. But the first thing that happens is water is split. Water is split. Oxygen leaves, okay, split, and we keep the hydrogen. However, the electron from the hydrogen is stolen from the little hydrogen. So we get an electron goes into PS2, and a proton gradient, or the hydrogen without the electron, proton gradient, builds up inside the thylakoid. Okay. Meanwhile, the electron is getting energized by free energy that comes from the sun. So this is available energy that the sun has. It's going to be bound up or excited, this electron. Now, don't ask me to explain exciting electrons. That's beyond my pay grade. Okay. So excites these electrons. Okay. And then the electrons are passed through the electron transport chain. When that happens, or the ETC, as we fondly call it, the electron transport chain, ETC, as they are passed down like a bucket brigade, the hydrogens, or protons, as we actually call them, the protons are pumped through using that energy from the electrons to turn on a little protein channel, active transport, move into the space, the thylakoid space, and build up a proton gradient there. Okay? The proton gradient that's built up here is then passed through the ATP synthase that turns on the turbine, which makes it like powered, a little bit of power, although this is passive transport because it's going from an area of high to an area of low. Doesn't require ATP to go or any sort of energy. It's just using the flow of the protons to go. Spins that turbine. When it does that, ADP, adenine di, meaning 2, phosphate, is attached to that phosphate to make adenosine triphosphate, 3 phosphates, okay? This is going to be used to power the next step of photosynthesis. Oh, here we are, back up here. Electron transport chain is just finished, and the electron then is going to be re-excited by more free energy from the sun. However, remember, PS2 powered up a little higher than PS1, and in what case those powered up electrons will be encased in NADP+, which is an electron carrier, converts that to NADPH because now it has, I should draw a little like converting arrow. Now it has the powered up electrons in there, and that can be used for the next step too. Okay? Do you see why I wanted to teach you this? So much. Okay? All right. How are we doing? Okay. So what we used so far, if you had your formula, you still have your formula, right? So hopefully. So what we use so far is in the light-dependent reactions, which we call LDR, light-dependent reactions, dependent on light. What we've used is water, so you could put LDR above the water, choose there, okay? And we have created oxygen, so you can put LDR above the oxygen, okay? So we've used half of our formula so far. All right, cool. We have ATP right here, and we have NADPH right here. Those two things are going to go over to the stroma. They're already in the stroma, but they're going to go and power the light independent reactions. Okay, are we ready for that? All right, before you take any quizzes on this, I need you to study it, okay? This isn't like a one and done thing. Like we work on this for like a week in class, like practicing and modeling and redoing. So do some studying before you start taking quizzes, okay? All right, I'm gonna erase. Anyone gonna die if I erase? Don't die. Cool. Okay. So thylakoid is somewhere else. Now we're in the stroma, which remember, that's the cytoplasm of the thylakoid. I am going to give you a very, very general description of the light independent reactions. We don't actually need to know as much about them. You can look up diagrams of them. I have a little diagram thing for you to do. We'll see if we get to it. Okay, light independent reactions, and this is AKA, otherwise known as Kelvin cycle. I don't think I want to tell you the other name too. It's got 3 names, citric acid cycle too, but really Kelvin cycle or light independent reactions. We used to call them the dark reactions. So you might see some places it's called dark reactions, but that's a misnomer. Like that's a bad name because they actually occur in the light too. It's not only in the dark. They just can work in the dark for a while, but they work during the light as well. So they don't have to be in the dark. So we call it light independent because they're not dependent on light. Okay, but Kelvin cycle is probably the name you'll see the most, named after a scientist that discovered it. Okay, so if you look at your formula, you'll see what we need, what we have left to do here. Okay, so we have carbon dioxide that needs to move in. So we haven't even dealt with that yet. Carbon dioxide enters the plant through little holes on the bottom of their leaves called stomata. So they actually kind of breathe in carbon dioxide, okay? And so carbon dioxide comes into the cell or comes into the plant and it goes to the stroma that is in the cytoplasm. That's like the cytoplasm of, oh my gosh, Miss Wilhoit, I apologize. Will you please take thylakoid off? Sorry, sometimes I get a little crazy. Not the cytoplasm of the thylakoid. That's a different name altogether. Cytoplasm of the chloroplast. We're in the chloroplast. We're in the middle of that big chloroplast, okay? And it has this cycle, okay? And so I'm going to draw a little cycle here. And like I said, I'm not going to get very in depth with it. There's 3 phases to this cycle. The carbon dioxide has to be fixed. So it has to be like changed. And so we have to go through like a fixation period. But basically what you need to know is that right here in the cycle, or early in the cycle, we'll say, we use some of our products. Okay, so we made ATP in the last part. We're going to use that. Okay, and we're also going to take some of the energy from our electrons. Okay, so we're using what we've made. That's basically what we're doing here. As we're doing that, it is taking a big molecule. Okay, it's taking a big, I'm trying to decide how much I really need you to memorize because this is a lot. It's taking a big molecule and we use, there's an enzyme called Rubisco that does that, but honestly, you don't even need to know the names. So basically it's taking a big molecule and it's converting it. It's using CO2, it's bringing carbon in. We're using the carbon from the CO2 and the oxygen really. And it's bringing that in and it's just converting it. So we're fixing it here. So we're changing it up, okay? And then we're going to, fixing is a weird word that you probably should know what it means. It means making it available for use, okay? If something is unusable and you have a chemical process that like fixes it, then the cell can use it. And so we're making the carbon dioxide available for use right here. And then down here, we have something produced called G3P. And it's basically a precursor of glucose, okay? And we actually have to go through this cycle multiple times to make an actual molecule of glucose. Okay, so it's like, this is the beginning of it, but this is the sugar. So this is the pre-sugar, okay? Pre-carbohydrate, pre-glucose, whatever you want to call it. Okay, and then over here we have to use some ATP again because we have to regenerate. So we have to regenerate our molecule. I'm not even going to tell you the name of the molecule. Half the time I get it wrong. It's huge. So we're going to regenerate our molecule to go through the cycle again, okay? And then more carbon dioxide comes in, and then it fixes, and then it uses ATP and ADPH, and then it makes this little pre-sugar thing, and then off it goes again. ADP plus P, ADP plus P, okay? And so then it uses energy to regenerate the molecule, and it just goes through the cycle over and over and over again, okay? What you really need to know about this is that it cannot go without the products of the light-dependent reaction. That is the power for this thing. So the light-dependent reactions create the power that does this Calvin cycle. And then what else you need to know is it's making a chemical energy out of this. So it's pretty cool. It's taking what energy was created in a solar energy type and making this chemical energy, storing a high energy molecule is what it's making out of that. And so basically what we've seen sun's energy go into edible energy, chemical energy. And so that's super cool. But what this does is it takes a big fat molecule, and I'll pull up a picture of the whole cycle here in a second so you can see the names of the molecule, but I'm not about to try to write them on here. So basically it takes this really big molecule, it fixes it. Takes a big molecule, brings in CO2, fixes it, goes through multiple series of steps, uses the energy from the light-dependent reactions, and then makes a little pre-sugar, which it can then bond to more pre-sugars to make the full sugar, and then uses a little more ATP to regenerate, okay? And then, of course, these ADP and the NADP plus will go back to the thylakoid to get recharged again. So like little school bus, go back and pick up the kids again, okay? Let me pull up a picture of it. I have actually a little thing we were going to do, but clearly we're not going to have time. But it takes you through and it asks you questions about different things. Like, so there's your little thing. And it asks you label the parts, do all of this. And so I'll stick this in your Canvas for you to do. I might just have instructions. It's called the pear deck. So, and then there's a little, helpful hints. But this is what the cycle looks like. Okay. So you have your 6-PGA, PGAL, and that's what I'm saying is you don't have to know all the names of all these things. And that's why we kind of have to find a weird happy medium for the Calvin cycle because it's got so much going on. So you can see some of these molecules that, like I said, I'm not about to try to, come up with that molecule for you. That's ridiculous. I had to memorize it when I was in cell bio, but we're not in cell bio. We're in intro to bio. Okay, so basically it goes through, series of steps, uses the energy molecules there, uses the energy molecules there, and then regenerates and does it again, over and over again until it runs out of the energy molecules because the sun went down and so they're not making any more. And so typically, actually, this can't run all the way through the night. It does stop. It does stop the cycle whenever it runs out of energy, okay? All right, so the Calvin cycle actually is much more simple than the light dependent for us because they make us memorize all the light dependent reaction parts and they don't make us memorize all the Calvin cycle. Thank heavens, good grief. Okay, questions? No, absolutely. Don't think this is, like I said, one and done thing. This is a week-long process in our class, modeling, memorizing, quizzing. I mean, it takes a while. So don't feel bad if you don't have it straight out the gate. It's not a big deal. It should take you a while to get it. And you only have 2 quizzes to do, and they're both over this. And so you could totally just study, and then take the 2 quizzes, at the end of the week or the beginning of next week. So not a big deal. I will also put this little pear deck up on Canvas so you can play with that. It has some questions, and then I'll put the recording on Canvas. And if I find any other fab resources, I'll put them up too. And maybe a nice blank drawing of the whole light-dependent reaction thing so you can try to fill in the blanks. Okay? All right. If you have questions, of course, you're always welcome to hang out and ask them. Otherwise, thank you for coming. I hope it was helpful. I hope you feel like it was nice to be taught for once. And I'll try to do this every Wednesday at this time for the foreseeable future, unless President's Week gonna have to be 1 o'clock again because I have kids in my classroom all day on President's Week that Wednesday. Okay. All right. Hasta luego.